All right. So, hopefully, we'll have some folks join us in a minute. In the meantime, allow me to hydrate. Okay. Let me just close the door real quick so I can minimize the sound coming from outside. In the meantime, if let's actually, I'll just wait. Okay, so I guess we'll get started. If you are interested in asking some questions, you know what to do. We have here an eye. I'm happy to go through this a little bit to give you an idea of the structure of the eye. And I also brought the brain home so we can talk about that a bit. I'll definitely take some time to go through the major parts that you need to know and kind of walk you through that a little bit. And other than that, I think I'm going to give folks about 2 or 3 more minutes to see if they can get here. And in the meantime, we'll just go ahead and put this up on the couch here. So, put your thinking caps on, and I'll be back in a minute. Okay. So I went away, and now we're up to 5. So fantastic. I'll grab my friend here, and we can get started. I guess I'll just go through kind of the major structures of the brain and the eye. I can't really get as much in depth with that particular eye model as I'd like to because, well, it's not letting me get in without breaking it. So we won't be able to see everything inside the eye, but I'll give you a pretty good idea of what we're looking at. So I figure I'll start there, and if anybody has any questions beyond that, we can certainly talk about them. Otherwise, if you're interested, if you're watching this right now live, there is an assignment that I'm going to start class with tomorrow and Wednesday called Finding Memeing Brain Edition. Again, if you have a better name, I'm all ears. Just a bunch of random GIFs and memes and a couple music videos on this one. I'm pretty proud of my work that are going to kind of jog your memory about different parts of the brain and what they do. So I think that'll be a pretty good use of our time to kind of review at the beginning of class. And then we will jump right into the review proper and go ahead and knock out a test. The test is 60 points. It is a mixture of multiple choice. It is some matching questions. It is a great big open response question focused on the functions that would be missing if certain parts of the brain and the eye were absent. So you'll have to forgive me if I don't talk directly about that tonight. and a diagram where you'll have to label parts of the brain and the eye. So again, no tricks. I'm not going to throw you any big curveballs here, but basically you're going to have to know your stuff. There's brain notes, there's eye notes, there's the test review with an answer key. I've actually got that open if anybody has any questions related to that. And that's about it. If you put in the work, then you'll be okay. So let's go ahead and start with the brain. I'm gonna get started. So we have here. Here's the back of the brain over here. This is a human brain model. So we notice that the cerebellum is sort of directly underneath the occipital lobe of the cerebrum here, and the brainstem goes downward and toward the back of the body. And so we have here, actually, I'm gonna go ahead and just use half of it here. We have here the frontal lobe, the parietal lobe on top, the temporal lobe here, which sort of has its own section. And we have the occipital lobe back here. So those are the four major lobes of the brain. They have the same names as the bones that lie over them in the skull. So those should be pretty easy to remember. Here in the frontal lobe, we have a lot of higher order thinking, your problem solving, your decision making. We have Broca's area right about here, which deals with the muscle movements involved in speech. And up here toward the back of the frontal lobe, right in front of an area called the central sulcus, we have the primary motor area, or the precentral gyrus, or the motor cortex. It has a lot of different names, and it helps to control all of the skeletal muscle contractions in the body. Then, if we look at the parietal lobe up here, the parietal lobe is all about sensory input. Right behind the central sulcus, we have the post-central gyrus, and that is an area that controls all of the sensory input. It's also called the primary sensory area or the somatosensory cortex. All of these areas have lots of names kind of based on what they do and also their location. The temporal lobe contains auditory centers, so all of the sounds that you hear are processed in the temporal lobe. It's just a coincidence that I was scratching my ear just then. And there are also olfactory centers in here that receive signals from the olfactory bulb or the olfactory nerves that are right here underneath the frontal lobe. So you can see how they sort of go back toward the temporal lobe, and deep inside the temporal lobe is where those olfactory centers are located. Then in the occipital lobe is where we find our visual centers. So the optic nerve, which I believe is right here, that is the root of the optic nerve underneath the brain. Obviously, this would branch out to both sides, and the eyes would be over here. So the optic nerves would go into the back of the eye, as we've discovered. And this would go all the way back to the occipital lobe, where those visual centers are located. And then we have another area. I'm actually showing you the right hemisphere, but both Broca's and Wernicke's areas are usually located in the left hemisphere of the brain. And Wernicke's area in particular, which is about right here, sort of encompasses part of the temporal, parietal, and occipital lobes all at once. It is the area that allows us to understand speech, whereas Broca's area allows us to actually form words. This right here is the cerebellum. It's responsible for coordinating muscle movements. And then we have all of the different parts of the brainstem. Up here, this sort of bluish area here is called the midbrain. Then we have the midbrain is actually sort of deep inside here. For the most part, actually here. If we look at the inside, the midbrain is sort of up here. Then we have the pons further down. The pons bulges out a bit. And then the medulla oblongata is here just before it sort of merges with the spinal cord. You'll remember the medulla is a really important area for automatic functions. Things like heart rate and blood pressure are centered there. The pons, which comes from the Latin for bridge, serves as a link between the cerebrum and the cerebellum as well as the spinal cord. So it helps relay impulses all through the brain and spinal cord. So now that we're looking at the inside of the brain, we have here in the center the thalamus, which is another very important sort of relay station for a lot of different impulses. It actually relays impulses in all directions, so to all different lobes of the brain, both sensory input as well as motor function can be helped along by the presence of the thalamus. And then we have the hypothalamus below it, this sort of triangle-shaped yellow area right here, and that is the center for a lot of homeostatic function, as well as an important part of the endocrine system that communicates with the pituitary gland. The pituitary gland would sit right below, I believe that actually is meant to represent the pituitary gland right here. And so there's a physical link as well as a functional link between those two areas. Then this section of blue tissue here, which was probably the hardest part for you to cut through during the brain dissection, that is the corpus callosum. And the corpus callosum is the set of longitudinal fibers, and that means they go back and forth, that help the two hemispheres to communicate with one another. And aside from that, that's about what you would need to know about the brain. You'll also need to know a little bit about the cerebral cortex. The cerebral cortex is the outermost couple of millimeters of the cerebrum, and that consists of gray matter, which is all cell bodies and unmyelinated axons. So things would tend to move a little slower within the cerebral cortex because of the short distances involved. That doesn't really affect things really profoundly. The rest of the brain and a good portion of the spinal cord and really all of our peripheral nerves consist of white matter, which is myelinated axons that tend to move things really quickly. So if I put this back together, just a couple important landmarks here. One obviously is the central sulcus, which is right about here. Whoops. It's right about here. That's the boundary between the frontal and the parietal lobes that has the primary motor area or the precentral gyrus in front of it, and the primary sensory area or the postcentral gyrus behind it. The other part that may come up at some point is this part right here. You had to break through this before you could cut through the corpus callosum when we dissected the brain last week. This is the longitudinal fissure. And a fissure generally is a deeper depression. So the sulci are these sort of shallow grooves here. The gyri are sort of these raised areas. And the more gyri and sulci a brain have, the more surface area there is for neurons and the sort of higher cognitive capacity an organism has. If you look at a rat brain, for example, not only is it really small, but it also is very smooth. And the smoothness of that brain, the lack of gyri and sulci, mean that it doesn't really get, that there isn't a whole lot of surface area for neurons, and therefore rats are comparatively kind of dumb. Sorry, rats. That said, there's countless examples of rats being trained to do certain things. I think rats have been trained to like drive little go-karts recently. So they're always trying to teach rats new things to see if they can kind of push the capacity of even their very simple brain to see if there are applications to helping us maximize the neural connections that we can make in our relatively sophisticated brains. So just kind of an interesting aside there. So that is probably as comprehensive a summary as I can make of the brain. Of course, if anybody has questions, feel free to chime in. That chat area is looking awful blank right now, and there are only 6 of you here. I was kind of hoping for double digits at least, but I will put the brain aside for now, and whoops. And I will go ahead and pick up the eye. Ooh, we're up to 7 now. 7. But first, hydrate. So, the next thing that I was thinking it would be a good idea to talk about is the eye. I think this should be the screenshot that I use for this live stream because this is staring into your soul. But all kidding aside, we've spent a bit of time over the past week or so talking about the eye. In most of my classes, we got to do a few simple sort of visual reflexes. We'll definitely do some more comprehensive vision testing when we get back from break. I think it's going to be a really fun, interesting way to come back from a long vacation. And I wanted to take some time to look at the eye. I want to kind of go over the major structures of the eye and what they do. Unfortunately, I'm not going to be able to show you everything with this model, but we can definitely talk it out. So the outermost layer of the eye, the white part of the eye here, is called the sclera. The sclera is made of really nice, tough connective tissue that helps to protect the eye, helps it to maintain its shape, and also has a limited number of blood vessels that allow it to sort of nourish the rest of the eye. Then in the middle, we have this transparent cornea. I'm actually going to take that out and kind of show you what it looks like here. So the cornea, if you can see it here, has this convex shape. It is curved outward, as you know. You know your eye isn't curved in, and that allows it to bend light toward the middle of the eye. So if light were to come in, if light were to come in right here, for example, it would go straight through. But if light comes in at any sort of angle, it's going to bend that light toward the middle of the eye. And the goal is to have all the rays of light converge on the retina. The retina is really where all the magic happens. So we've talked about the sclera. We've talked about the cornea here. Now, let's go a little deeper. So if we look a little further here, and again, I can't really get any further than this. This darkened area is called the choroid coat, and you may notice the red and blue are blood vessels. So there's obviously a lot of nerves here that help to send impulses throughout the eye, but there's also these blood vessels, and the choroid coat, aside from having this dark color that absorbs light very efficiently to kind of get rid of the noise that might come in, the excess light that may make it harder to focus on objects. It also has blood vessels to help provide oxygen and nutrients to all of the tissues of the eye. So it's a really important layer that we don't talk about that much because it's in the middle. So the choroid coat is really important because it's what we call the vascular layer of the eye. That is, it has a whole bunch of blood vessels. In the cow eyes that we saw that the choroid coat actually had about half of it toward the back of the eye was actually very bright. That was the tapetum lucidum, which is a bright layer that instead of absorbing all the light, which something that is black would do, it actually reflects a lot of the light back onto the retina. And that's what not only causes their eyes to appear really bright at night when you shine a light on them, but it also helps them to see better at night because more of that light that normally would just go right through is reflected back onto the retina so they can translate it into an impulse. So now what we're looking at here is the iris. So you can see, if you look closely, you can kind of see that sort of ribbed pattern of muscles there. There are actually two sets of muscles that are oriented in slightly different ways, and their job is to control the shape of, not the shape, but the diameter of the pupil. The pupil is always going to be circular like this, but it almost like a camera. It's going to open and close and get bigger and smaller according to the action of the muscles of the iris. If you have a lot of light coming into the eye, the pupil tends to constrict. We saw that last week when we did a quick demonstration. And when there's not a lot of light, your eye wants to open up nice and wide, and the pupil gets dilated so that as much light as possible can come in. Um, when a doctor dilates your eyes, as we talked about in class, they're basically paralyzing the muscles of the iris that would normally cause your pupil to constrict when a lot of light shined in. That way, they can shine their very bright light and they can get a very good view of the retina and make sure everything's healthy. So, I can't actually show you the retina in here, but you remember that the retina in the cow eye was sort of a yellowish layer. And that yellowish layer looked pretty ordinary. There wasn't a whole lot to it, but the retina is actually the most important layer of the eye. The goal of vision is to focus rays of light specifically on the retina, and even more specifically on a very small area of a high concentration of photoreceptors called the fovea centralis. So when an eye doctor, and I have two downstairs, one who's living with us for a while, and the other of whom I've been married to for 15 years. The goal of an eye doctor is to correct your vision so that the rays of light that are coming into your eye, they converge on that specific spot, on the spot with the highest concentration of photoreceptors. So if you're myopic, if you are nearsighted, that usually results from either an inability to curve rays of light as much as they need to. Actually, it really results in sort of an over curvature of light, so the rays converge too soon. And it can result from sort of anterior and posterior segments of the eye that are too long. And so what they want to do is they want to curve those rays of light outward, and so the lens that you get is concave. It is sort of thinner in the middle than on the outside, so that the rays curve outward. Because the problem in myopia, in nearsightedness, is that the rays converge too soon. If you're farsighted, if you have trouble, if you can see at distance with very little trouble, but if you have trouble seeing things close up, the solution is to put in another lens that's much like the cornea and the lens of your own eye, that is convex, that is bigger in the middle and that curves things further in. Because the problem in hyperopia or farsightedness is that the rays would converge at some point beyond the retina. So they really never converge. And so the goal is to kind of bring them back, to get them to curve a little bit further in, and to converge sooner than they normally would. Um, so, um, so the two types of photoreceptors that we have are rods and cones. Rods, way more abundant. You have a ton of them. They allow you to see in low light because they're really sensitive to light. It doesn't take much light to activate them. But they only really allow you to see sort of general outlines and shapes. So there's not a lot of detail there. The cones are where you get your detail. There are three types of cones: red, green, and blue. They help you to see all of the color and all of the details that you would need to see. If you're colorblind, you're missing one or more types of those cones. Um, Most animals don't have cones, so they can't see color. So which is kind of an interesting thing. So let's see. Talked about rods and cones. Um, a lot of the details that I gave you in the eye dissection packet um, are not necessarily going to be covered here. I think if you understand the basic parts of the eye, the ones that I covered in the notes, the ones that had you identify in the dissection, you should be more than good to go. I'd love to go into more detail, but in the interest of time, we had to kind of streamline things a little bit. And aside from that, the only other thing of time we had to kind of streamline things a little bit. And aside from that, the only other thing about a whole lot is dopamine. It's an excitatory neurotransmitter that really focuses on the pleasure centers of your brain, the parts that are activated when you're doing something that gives you a lot of pleasure, and they artificially increase the levels of dopamine through various means. Sometimes they shut off transporters, which would take dopamine out of the synapse. Sometimes they increase the production of dopamine or the ability of the presynaptic neuron to dump it into the synapse. They do it in a lot of different ways, but the end result is you feel really good for a really short time. And then because your neurons sort of start forgetting how to make those important chemicals that make you feel really good, the only way to get those again is to take the drug. I've ranted about nicotine a whole lot, and the reason that adults are so concerned about this new vaping epidemic that we have, aside from the fact that people are getting black market stuff that rots out their lungs, and we don't really understand why, is the fact that nicotine is incredibly addictive. It has about half the effect of heroin in terms of the increase in the levels of dopamine. It increases the levels of dopamine in those pleasure centers of your brain by about 3.5 times. No wonder it makes you feel really great when you vape. But it is also incredibly addictive for that reason. So once you start, it's really, really hard to stop. Um, and I'm encouraged always by the way that addiction is treated more as a public health problem, because it is exactly that. It causes health issues for people, and the addictive aspect of it makes it really hard to stop. So I think that's about all I got. Usually I want these to be a little more interactive, but at least I could talk about this for half an hour. If you got any questions, I'm more than happy to answer them. And if there aren't any questions, I might just go and eat my dinner because dinner wasn't quite ready. We tried, but we failed. And Emma Brissell just sent me a really cool resource that actually is about at the level that I want us to be at, which will allow you to practice labeling the eye. So thanks, Emma, if you're out there, for sharing this with me. I'm going to go ahead and send that in an email. It's just a really cool online tutorial that allows you to kind of label the parts of the eye, and you can compete to see who does it the quickest. So I'll pass that along. I'm going to step out for a minute. I'm going to give you a chance to ask any questions that you might have. Feel free to email me. Feel free to hit me up in that chat box on the side of the screen there. And otherwise, I'm going to call it a night if that's cool with you. So I'll give you another 2 minutes to kind of put things in there, and then we'll see where we're at. Thanks. Okay, so I guess we're good then. So look for that email if you're interested in yet another resource that will allow you to kind of label the parts of the eye. I think that the one that Emma found actually fits really well with what I would like you to do. Don't worry, you do get a word bank. So, if you have any questions in the meantime, I'll be at school in the morning. You can always ask before we start, and we'll have a little bit of time to review. And I actually have a specific activity in mind for that. So, hopefully, this has been helpful. And since I don't see any questions, I'm going to go ahead and sign off. Maybe I should just start recording these ahead of time in the future. But hopefully, this helped. Hopefully, the 6 people that are listening right now are a little better off than they were before we started. And I guess I will see you tomorrow or Wednesday. If you have any questions, feel free to hit me up. Have a great night.